Welcome to today's webinar. I think this is our first since October, so thank you very much for joining us. As ever, I would like to thank very much our sort of supporters, which is the British Society of Proteome Research and the London Biological School of Mass Spectrometry Discussion Group, London Biological Mass Spectrometry Discussion Group, who we always like to give a shout out to, as well as the London Metabolomic Network, the sort of 3 mass spectrometry related discussion groups are in central London, and also thank you to Imperial for giving us the hosting privileges for the webinars. I'd like to welcome everyone. So there's quite a large audience hopefully today, lots of people registered and from all over the world, which is a really nice thing to see. So thanks very much for continuing to join us, and for everybody on YouTube who watches these afterwards, it's all really greatly appreciated, and we're glad that they make it out to such an audience. If anyone has any feedback ever or speaker suggestions or would like to speak, please do get in touch and let us know. We're always looking for new presenters as well as new committee members. And if anyone from anywhere in the world is interested in joining our committee and helping to run the webinars or our seminar series in London, then please do get in touch. We're always really great to receive new members. As normal, if you have any questions during the webinar, please can you put those into the Slack channel. The link to join it is on the screen at the moment. If you do put any questions into the Q&A on Teams, we will transfer those to the Slack as well, so they'll all be on there. We have 2 really great speakers lined up today, Eneko Villanueva and Georg Borner. And today's guest chair, Professor Helen Cooper from Birmingham. We're really proud and pleased that she can join us today. She's a EPSRC Fellow at the School of Biosciences at the University of Birmingham, and her research focuses on developing mass spectrometry methods for in situ analysis of proteins. The aim is to combine ambient mass spectrometry, ion mobility spectrometry, and native MS to obtain spatial and structural information about proteins and their complexes directly from the physiological environment. She's director of the Rosalind Franklin Institute and serves on the editorial board of the Journal of the American Society for Mass Spec and Analysis and Sensing, and to cap it all off, she's also a fellow of the Royal Society of Chemistry. So we're really very pleased and proud that she's able to join us and do the guest chairing today. And with that, I will hand over to Helen to introduce our first speaker. Thank you very much.
Thanks, Harry. It's a real pleasure to be here today and to be chairing this webinar on spatial proteomics. As Harry says, we've got 2 great speakers for you. The first of which is Eneko Villanueva. Eneko is a Wellcome Trust postdoctoral fellow in the Cambridge Centre for Proteomics. He studied biology at the Pompeu Fabra University in Barcelona and then went on to complete a PhD at the University of Barcelona. In his current role, he is developing new technologies to understand the crosstalk between proteins and RNA. And his talk today is titled a system-wide quantitative map of RNA and protein subcellular localization dynamics. So, Eneko.
Yeah, perfect. Thank you very much for the introduction, and thank you very much to the organizers for giving me the opportunity of being here today presenting what has been my work during some years now. So, since I'm the first speaker, I assume that I will need to introduce why to study protein localization inside of the cell. But first, just yes, I have to say that this has been a massive project involving 2 different groups, one of them in the University of Cambridge. Well, now both of them at the University of Cambridge, one of them in the Department of Biochemistry, the other one, the Mass Toxicology unit, with a lot of people involved in it, and I'm just a representative. So, when we want to study how the cell is organized, I mean, for many of you, it will be as easy as take a look at it under the microscope. But if you want to go deeper and see how the different organelles behave inside of the cell, you want to start to have a more complicated approach. And if once you want to go really deep and take a look at how different macromolecules are organized in the cell, I mean, for the DNA is quite easy. It's just in the nucleus, but the topic of today is trying to understand how the proteins are distributed in the cell, and importantly, also how the RNA is organized, so trying to obtain a very multi-omics view. So let's start with proteins first. I mean, for many of you, you have a targeted question. It's always better to go for a targeted method. So if you're interested in knowing where few proteins are, where some proteins are, you can directly immunostain them, and this is absolutely the way to go, unless you really want to do it in dynamic conditions, many different cells and many proteins at the same time, where things start to be complicated. If you are interested in a given compartment, what you can do is you can engineer your cells so they can express apex proteins to biotinolate proteins surrounding a protein of interest. And if this protein is really well localized in a given compartment, you can obtain enrichment of protein localization. Sorry, this should not have happened in the periphery of a given organelle. But again, you have to engineer your cells. You have to express it in a given compartment at a time, and this, I mean, even if it's doable, is quite challenging for dynamic conditions. And lastly, a more classical approach will be just to enrich the compartment of interest and check which proteins are there. We expand this concept, or we can expand this concept at least, and instead of purifying organelles, what you can do is reorder the entirety of the cellular content, and by analyzing the proteins that are along this gradient, what we can retrieve are these correlation profiles. And this is probably the key idea of this talk, is that you don't require purification, you just need signatures of proteins that belong to different organelles along any kind of gradient. In here it could be density, but we will talk about different approaches later. Later on, any protein that shares exactly the same kind of signature can be classified to belong to the same organelle. We call this methodology localization of organelle proteins by isotope tagging, even if I think that we have to update the name because it doesn't require necessarily isotope tagging. You can use any mass spectrometry quantitation methodology you want. Importantly, during the years, we have expanded this concept to not only know which proteins belong to a given organelle, but also to give some numbers about them. So we can really know which is the probability of a given protein to really belong to a given organelle, or even to be shared between different compartments, as you can see here. And this is especially important for proteins that are traveling between compartments, as do the great majority of the proteins. Or even put it further and analyze how proteins change in dynamic conditions. And this technology allows us to have extreme precision, not only in the proteins that change inside of the different compartment, but even inside of the different compartments with different probability. So this allows us to put very stringent thresholds of what we consider movers and what we consider stable proteins. And as I assume everyone will say always, our method outperforms any other analysis method. And if you are interested about it, go to bioRxiv. The paper is going to be out very, very soon. But in the meantime, you can read about these analysis tools in this preprint. If you are interested, generally speaking, more about the LPD technology in general, go to PubMed. There are plenty of publications currently. And if not, just drop us a line and we can assist you how to implement it in your models. But this is a multi-omics talk, so we are interested not only in proteins, but also in the RNA. And the reason is that we want to understand how the cell is organized, not just how proteins are organized. But considering the audience, I think that it's also quite important to remember that many different proteins can use RNA as a scaffold. So many proteins are localized to RNA, and we will talk about it a little bit more in detail. But lastly, why to study RNA? I mean, mainly because you have to think about your parents. Every protein has been an RNA before, and knowing what happens with the RNA gives you an idea of what is going to happen in future to the cell. So now a little bit more seriously, I think that many of us will have to reconsider, and I'm the first one that I have to reconsider, what are cell complexes, because we know nowadays that many of them contain RNA. Of course, if I told you about a ribosome, the first thing that will pop on your mind probably is that it has RNA, but it has tons of proteins in it. The RNA polymerase, of course, is a complex between RNA and proteins, but even other complexes like the SRP, that is a protein complex that drives the nascent polypeptide to the ER, contain non-coding RNAs on it. And we know that many proteins are interacting with RNA or are localized to the RNA, mainly because recently we published this other technology that is called orthogonal organic phase separation to retrieve RNA binding proteins. It's very, very straightforward. You have to stabilize RNA protein interactions using UV light, and then you just do a normal phase separation, trisol, a commercial reagent, in which RNA goes up to the aqueous phase, proteins go down to the organic phase, and crosslink samples, just sharing physicochemical properties of both RNA and proteins, are trapped in the interface. Then if you are interested into the RNA that is interacting with proteins, digest the protein. If you're interested into the proteins that are binding RNA, just digest the RNA, and proteins go back to where they should be, that is the organic phase. So if you crosslink, proteins are enriching the interface. If you treat with RNases, proteins migrate down to the organic phase. And what this technology allowed us to do some time ago now is to retrieve the total RBPome. So not only proteins that are bound to mRNA, but proteins that are bound to any RNA. And as you can see here, around 1,500 proteins, very abundant proteins, many of them are bound to RNA. This means that if your localization method is not able to properly allocate RNA in the cell, you may be missing the localization of many proteins. For the ones of you that are interested about how to recover RBPs, just go to these manuscripts. And going back to localization of this multi-omics approach, so RNA and protein, mapping the protein is quite a well-established field, as I already explained to you. Mapping the RNA should be easy in theory, because protein are localized to many different compartments, but RNA is not localized in so many places, so it should be simpler. Also, the chemical space of the protein is quite diverse, while the RNA is quite a simple molecule in comparison. And I mean, we all know about the different foldings of proteins and what is very, very diverse. What happens with the RNA is that it's very dynamic, they're folding, and something that is extremely important is that it's a massive molecule. So the dynamic range of RNA is absolutely massive. We can go from microRNAs to very, very long RNAs. So every RNA that codes for every protein is longer than the protein that is coding to, and this has strong implications for method development. So, generally speaking, just few slides like what happens with proteins. If you are interested in a given RNA, do FISH, just synthesize some proofs that hybridize with your RNA of interest and take a look at it under the microscopes. This is absolutely the way to go. But with like immunofluorescence, if you are interested in many different RNAs in different conditions, different cell lines, this can be complex. If you're interested in a given compartment, yes, the RNA can be biotinylated in the same way that proteins can, and then you can follow exactly the same approach, but having exactly the same limitations. Like, for example, open compartments are usually challenging with this kind of approach, and dynamic conditions are also quite demanding. So imagine different compartments in different conditions. What happens if you want to interrogate all compartments at once and all transcripts at once? Well, this kind of idea has been previously attempted, and the great majority of the people set up their methods using proteins first. So something that can work for proteins, like for example in here enriching membranes, doesn't necessarily work for RNA, because as I told you, RNA are massive molecules, so big that sometimes sediment in a pattern similar to organelles themselves. So this kind of issue of the RNA size co-sedimenting with organelles, giving some kind of data difficult to interpret, let's put it this way, has been common in many different approaches. Like for example, this other one trying to recover the protein on subcellular localization, that what happens is that in using very high speed centrifugation, very long RNAs are depleted from the later fractions. As a result, there are almost no RNA and therefore no RBPs binding these RNAs in membranes. Or even more recently, so for example, here in the cytoplasm, protein and RNA localization don't agree, meaning that one of them is not properly allocated. So with all of this in mind, we thought, okay, maybe we can attempt to do it using our correlation profiles, so our signatures, to not only obtain maps of where the proteins are in the cell, but also maps of where the RNA is in the cell. This was some time ago when I started this project. Pre-COVID, things were different. But what is important is what we use our gold standard method at that point, that is hyperlopit. And first you enrich for membranes, then you run these membranes inside of a gradient, and then we quantify RNA and protein there. And the problem is that we had no RNA whatsoever in the cytosol. So obviously this was terribly wrong. And what I've been doing during this year is, let's say, like an exercise of minimalism. We have been trying to trim our approaches until a point in which there are so few steps that it's almost impossible to fail. Let's put it this way. So I will skip this one because it's quite technical. You have everything in the manuscript that I will link later on. But just a very side thing for anyone interested in method development, keep in mind that it may take a little bit longer than what you think. Okay, now the method. So it's extremely straightforward, as you can see. So we have gone down 10 times with the cells that were required using previous attempts. And the only thing that you need to do now is to lyse your cells. Of course, you have to reorder it, load them inside of a density gradient, go home, sleep, and come back the next day to the lab once the cell has been completely reordered according to the density of its constituents. Then recover RNA and protein and do sequencing and mass spectrometry analysis of the fractions. Let's start first taking a look at the gradient. This is the anatomy of the gradient. These are the usual Western plots that you have to always show, showing that you have potential resolution. But let's go deep and take a look of what happens when you put the fractions in the mass spec. So seems that, or indeed it is the case, that different organelles have different correlation profile, which allow us again to have this kind of maps of where the proteins are. If you prefer in a higher dimensionality space, this is a TSD with proteins classified to the different compartments with quite high resolution. But this was the minimum. We were already able to do it. The key question is, can we go from proteomics to multiomics? So if we take the RNA that is coming from exactly the same fractions along the gradient, what we can see is that RNA that is known to be in different localization in the cell also have different correlation profiles, what allow us to have, for the first time ever, simultaneous maps of all RNA localizations at the same time. This means nucleolus, nucleus, cytoplasm, ER, mitochondria, and a different subcytosolic cluster that I will discuss later on. Something very important that I want to remark quite a lot is that since then we have put in here the entirety of a cell lysate, nothing is out from here. So this means that we can correct, and instead of having relative profiles, we can have absolute profiles. And this allows us to quantify exactly which is the proportionality of macromolecules in every given compartment, and going from relative profiles like these ones to absolute profiles of which is the percentage of a given molecule in every given compartment in the cell. So you can imagine that at that point we were quite satisfied, we were quite happy with it, but the problem is that I am the wet lab scientist. And I felt a little bit more like Pandoran here, because, and this is a personal belief, I think that when you develop a method that is high throughput method, you can justify that everything is validated just using few proteins or transcripts. Usually you have to develop another high throughput method to validate the first one. So we did. So what we use is a different kind of lopit approach. The ones that I've been talking to you about use density to retrieve protein or RNA localization. What we did now is use different sedimentation speed. So in this case, you take your cell lysate and you spin it down at different times, take the supernatant, put it in another Eppendorf, spin it down, and every pellet is going to be enriched in different compartments of subcellular localizations. But remember, I told you before, the RNA is massive. So again, with a lot of effort, we just simplify it again until a point in which we were able to obtain extremely good subcellular localization of RNA and protein just using 4 different centrifugations and 5,000 Gs, which means that everyone can do it in a benchtop centrifuge. And what is important is that we have done it avoiding any kind of RNA size bias effect. So when we compare our density methods to obtain RNA and protein subcellular localization with the sedimentation-based methods, what we can see is that they agree remarkably well. So these are the transcripts that were in membranes, and they go exactly to the membranes in the other one, nucleus and cytosol. But even the proportionality is extremely accurate between both methods, so they agree remarkably well, even considering that the molecules or the organelles are reordered using completely different physicochemical properties. So let's see what this is able to tell us about cell biology. First things first, for example, long non-coding RNAs, this should be an easy one. It has to be spread between nucleus and cytosol in the cell, and indeed it is, but we found that the long non-coding RNAs, the more abundant ones, are highly exported to the cytosol, and we model the features that drive this export, and this very simple 3 features model outperforms, or at least is it as good, predicting export as models containing over 80 different features. But what happens with the transcripts that code for proteins that are going to be later on localized to organelles? So if they have transmembrane domains of signal recognition peptides, of course, they are highly enriched in membranes, but even some of the transcripts that do not code for canonical membrane proteins are allocated to the ER. One of the classical examples that I always like to talk about is this SRP alpha subunit that has a co-translational localization that I think that is quite cool. As soon as the protein starts to be translated, it docks to the subunit beta and translation continues in situ. So these are the kind of events that we are able to see using this technology. And if we take a look of all these transcripts that are located into the periphery of the ER, what we can see is that many of them code for cytoskeletal related functions. We validate 2 of them, not only about their localization, but about proportionality, and we were absolutely spot on. We'll come back to the cytoskeletal RNAs later on in the talk. But I've been talking about this in many places already, and I know, and I assume that in here is going to be quite similar, that the audience really want to know about this other different cluster without any kind of known RNA or protein marker there. So this was discovered using unsupervised clustering, what it is. And many times when you need to understand the system better, the best thing that you can do it is just challenge it a little bit. So what we did is we disturbed completely calcium homeostasis in the cell, and this generates some folded protein response that at the end what does is phosphorylates the initiation factor to alpha. This means, in a nutshell, that the polysomes go down to almost nothing, so it means minimal translation, and RNAs are trapped into granules. And since the method is so simple, we decided to do it many times, and many times, and more times, and more times. And I know that when talking about this kind of gradient, some people can have concerns about replicability, but since this is a single step approach, the replicability is quite easy to achieve. So yeah, starting with proteins again. So we are challenging protein synthesis mainly in the secretory pathway, so it's not a surprise that Golgi is the most affected organelle, but we found some proteins behaving in a little bit unexpected way, like for example some proteins leaving the ribosomes in here, having this pattern, and going to the ER. And indeed, we validated and we believe that this ZNF622 protein, what it's doing is removing the transcripts from the ER to the cytosol or to somewhere else that we will discuss later. But we also use our proteomics to inform our transcriptomics approach, because everything comes exactly from the same fractions. You can transfer the learning you are obtaining from one omic to the other. And for the ones of you that work in translation related fields, you will easily recognize these proteins. These proteins are usually going to the stress granules upon stress. And for the ones of you that cannot read matrix and interpret easily these kind of profiles, so these proteins are in here, part of this translation clustering here that has all the ribosomes. And as you can see, they move together to this other part in the cytosol. And this is described, or mainly described, by this specific fraction containing the cytosol light. And indeed, to this green cluster that is the cytosol light is where a lot of RNAs that were before in membranes or in the cytosol, after stress, they are going to exactly the same fraction. So in a nutshell, what they are, well, we discovered that our cytosol light are indeed granules. So we have been able to stabilize granules of RNA and proteins inside of this density gradient, what means that we can interrogate them now. And what we found is that the RNAs that go there are RNAs with very low abundance of ribosome occupancy and very long, both things, normal features of RNA that go to granules. And we also discover which are the proteins that are bound to them. But very importantly, since we knew the localization of the transcripts before and after the stimuli, what we were able to retrieve or to quantify is which transcripts are recruited more efficiently to granules. Unsurprisingly, we found that membrane RNAs are recruited more efficiently. And this is a surprise because it's known that membrane RNAs are not the main constituent of granules. But what we found is that the ones that go there do it very, very efficiently. And indeed, they do it in a sequence-dependent manner, and yes, the codon and with a very specific codon usage. Just to finish, almost, so which other things were we able to interrogate? I mean, we took a look of which transcripts were able to still remain attached to the ER. So this is before the stimulation. All these transcripts are attached to the ER. This is what happens after stimulation. And what we found is that they were, almost all of them, able to be bound by a different translation factor, not the one that is phosphorylated and stops translation, but the 3D factor. And indeed, they are the same transcripts that I told you before that code for cytoskeletal proteins. And by knocking down this factor, what happens is that the cell is not able to remodel the cytoskeleton anymore, is not able to move, and is not responding well to the treatment. So, in a nutshell, we have developed indeed two different methodologies able to retrieve RNA and protein subcellular localization at once and be able to obtain absolute quantitation of the macromolecules inside of the gradient. Discovered that 3D, the initiation factor 3D is required for cytoskeletal function upon UPR, and model that membrane localized RNAs are recruited more efficiently to granules. So now you will be, so if you are interested in this data, we have generated a user-friendly exploration tool in order to take a look of all our results. And if you want to know how to implement this kind of technology in your lab, you can scan this QR code with your phone or directly go to bioRxiv and take a look at this manuscript where we detail how we develop the method and how you can implement it. Just a teaser. We have not stopped there, but this is going to be for a different webinar. So you can expand this concept of correlation profile to not only obtain maps of RNA, but also of RNA and protein, but also the combination of both and have functional maps of proteins holding RNA in different places. Well, this was it. Thank you very much to the organizers and for all the teams that has been working on this.
Thanks very much, Eneko. That's a fascinating talk. Let's move over to some questions now. If I just look in the Slack channel, let's see what we've got. Okay, so we've got a question here. How do you allocate RNA to each compartment? Which RNAs do you use for each to train the clustering? So we have done two different approaches. One of them, for the ones that we have known markers to belong to a given organelle, we have used them as markers. So RNAs that are usually highly enriched, like for example, RNAs that are never exported, so they are classified as nuclei. In mitochondria, RNAs that are encoded by the mitochondrial genome. For membranes, we have used in situ biotinylation of transcripts that are translated into the ER
as ER transcripts. But we have also used unsupervised clustering to try to dig deeper into the data set, and this is how we found that something else was going on there and described the granules. So it's a combination approach. You have everything in GitHub. If you are interested in knowing it in detail, just go to the manuscript. It will link you to GitHub, and all the data analysis is available there.
Thank you. So another question, do you find any post-translational modifications that correlate with protein RNA complexes?
That's a very good question. We have not gone so far, but it's absolutely doable. So you can take a look at these proteins or even RNA and use just a phosphor enrichment or any other PTM enrichment and take a look if there is any kind of correlation. We have not checked it.
Okay. Can you calculate stoichiometries between proteins and RNAs? Do you see the same RNA proteins in both bound and unbound states?
That's again a very good question. That's very straightforward for RNA. It's not so straightforward with proteins. We have not been able to do it now. Absolute profiles for proteins are way more challenging. So the thing is that it's very easy to use spikes for RNA. And reconstruct, and since you almost don't have any bias into the precipitation steps for RNA, so it's an easy molecule in this sense, you can reconstruct very easily. For proteins, it's a little bit more demanding, and I can only say that we are working on it. So in not a long time, we will manage to do it.
Okay, thank you. So there's one last question. What about the proteins trapped inside vesicles, such as endoscopes and autophagosomes? Is there any way to detect them?
It can be. So the thing is, we had to take some compromises in here. So using other, in order to make it work with RNA and proteins, and in reality, this was almost a fishing expedition. So we tried to do it as best as we can, but this was the very first paper able to do it. We went a little bit in a conservative way, trying to recover mainly the main localizations or the ones that are easy to retrieve. If you want to go deeper and have sub-compartments, or even you will need to do mass spec in even more fractions along the gradient. So it will involve a little bit more expensive experiment. It is doable, absolutely. It was not the aim of the manuscript.
Okay, thanks very much, and yeah, thanks for your talk, Eneko. I think we'll move on now to.
There we go, we're in. Thank you very much. I think that was a really fascinating webinar. Thank you, Georg and Eneko and Helen for sharing. I've only got a couple more announcements to finish off. That is that if you haven't already seen, BSPR have announced their lineup for their annual conference this year, which is going to be an in-person event, as these are now being allowed again, which is really exciting. Check out their website for the details on that. It's in Oxford. And also LPDG, we're also having our first in-person seminar in March. Please check out our website for that, and if Jack could move the slide on one, you'll see a little advert for exactly where it is. Perfect. And I think that's all from us. If you would like to get a certificate of attendance, please, Jack will flash up the QR code. We'll leave it up for a minute, and that form will be there for around about an hour, I think. You'll be able to register for one of those. So once more, thanks very much for everyone for joining us, watching. We've put all questions on Slack channel, and I know Eneko and Julia from Georg's lab have been on there answering. So please, if you have any more, please do interact on the Slack channel. And once again, thank you very much everyone for joining and listening.